Let me introduce
Please.
you myself a little bit. I'm from IRTA at Catalonia, and I will present, as I was asked by Livia Lozano to do, a little speech or talk about the in vitro culture in what could be used in organic breeding. Actually, let's see if I can show you the presentation. I'm not an expert on Zoom presentation, so I'm a little bit confused here with these menus. Sorry about it.
You're not the only one, but we see the presentation in full screen already.
Ah, you do? Ah, okay.
Yes. Yeah, it's
perfect. That's perfect. All right, then I don't see it and you do. It's amazing. Well, let's see.
But we don't see the first slide.
Actually, I don't see anything.
Ah, okay, that's a problem. Okay, we see your screen.
Okay, now, probably.
We see the desk with the mountains.
Mountains and a lake and okay, yes,
now,
yes,
probably.
yes, that's good.
Okay, we are here in Catalonia, so that you get located. We produce quite a lot of fruits here, as you know. I'm a researcher in the plant tissue culture lab that has a 30-year-old history. So it's about a long time to do a lot of things with different crops, like all of those that you see in this slide. Okay, and the in vitro culture, as I started when I was young, it is conceived as a tool for the breeding of all any kind of crops, for each one of them with different purposes. And the techniques that are written here in the center of the slide are the ones that are probably have taken most of our time, and micropropagation and cleaning the plants from viruses and so on. So is one of the main works that we do. The embryo rescue of those crosses that are not viable is also important too. And then the production of pure lines or double haploids, which in fruit trees have not been solved as much. Of course, as in the vegetable crops, because these ones are propagated by hybrid seeds. But in the beginning of the development of these techniques, it was conceived also the possibility of doing double haploids of fruit trees, as I'll show you at the end. The polyploids are also important. Actually, some pair varieties are tetraploids, so as they were selected in the field by their phenotypic characters. And the cell suspensions that I want to talk to you about is also a soilless and actually a cropless plant production. And I will miss this, and as you can see here, I'm not talking about any genetic transformation, because we have not been involved in these processes during the last, I would say, 15 years or so, because the private companies and the growers, as you know, they don't want to deal with fresh and vegetable fruits and from transgenic plants. Well, and I actually I had to look at what would be the main characteristics for an organic plant breeding, and you could probably correct me, but producing new varieties under these conditions is quite a big effort, because at least in the area where we are, the organic farming is just a small percentage of the production. And from the course, I look at which were the main topics that you were concerned about, and I identify three in which the in vitro culture could do something. such as the collection of and management evaluation. Well, provide material for those assays. When one, as I heard before, is selecting a fruit tree and it doesn't have as much budwood, in vitro culture could accelerate the propagation of that material. There will be concerns about juvenility and so on, but at least you could produce thousands of plants in 1 year of work. Then the other topic is the genetic diversity exploitation, because actually the genetic diversity exists, but if one doesn't have the tools to produce much more plants, conserve them as at the same time as they are evaluated in the field, it has the risk of losing its work. And in vitro can provide a bank of germplasm. And then the evaluation for biotic and abiotic stresses, as you will see, has been also an objective in our group, at least in pears and apple, as you will see. Okay, so all the presentation is about propagation, sanitation, which I would tag with the yellow post-it, I would say. And this makes the sense is to take profit of the existing selected genotypes. We'll propagate them and keep them safe of viruses and diseases. And if they are contaminated, we could get rid of the viruses after establishing the material in vitro and propagate this one. The green tags are the selections of the materials in vitro. It can be done for some things, and in the future it will be done much easier. After I'll show you a tool that we have developed at IRTA. And the rest is to generate more genetic recommendations. And in vitro, it helps the rescue of embryos that would not be viable. They would not germinate in soil because they are immature or because they are interspecific hybrids. Let's go with an example of an apple plantation that had some Armillaria infection in the soil, and it naturally eliminated all the plants except 2 trees. And how could you recover the root stock from those trees unless you do something like what we have done in some years ago, that was collecting the roots, the plant that you see here sprouting. I don't know if you see my pointer. You do here. In this, is this a piece of root? And then it is sprouting. And from these shoots, we introduce them in vitro and propagate them and root them and bring them into nursery trial. So it showed, as long as the pathologists have told me, some tolerance to Armillaria. And actually, we haven't gone on with it because we are involved in so many crops that we didn't go on because this plant material had problems of propagation. Even though you see here some rooted plants, is different from having thousands of rooted plants, which is what the production requires. And the same for some walnut varieties and pistachio, self-rooted. When we had some trees located that had a good production, we wanted to evaluate them as a rootstock and also as a nut producer. These are rootstocks of peach, some that were identified, and then when the breeder comes to a point in which, as we said before, the plants are not easily propagated by cuttings, they ask us to solve the problem with in vitro propagation. Okay. I'm not saying with this that we solve all the propagation problems. No? I'm only saying that in some cases we are successful up to some point. This is a Intensia, which is a peach almond rootstock that has been selected under field conditions, and then we propagate them in vitro to produce more, a higher amount of plant material. These are the trees, which is a nice one shoot growth straight, which is a quite interesting rootstock. Excuse me. They grow in the greenhouse, of course. And the in vitro culture has the advantage of shortening the quarantine requirements with when the plant material is shipped abroad. And we have sent it from our facilities to, as is seen with the green arrows, to different countries. And we have collected plant materials, and the in vitro lab is also the receptor of those plant materials. Of course, one has to realize that before it's moved, the sanitary authorities of the country that receives the plant material has the guidelines of which plant material should be, which diseases should be free. And then once we have analyzed it and it's certified, then we ship it this way. The same thing for pears and cherries for wood production in forest plantations, or even for a F2 population of melon, when each individual is different and has to be propagated because it has to, the reader wants to evaluate the genotypes in different sites. This is a better convenient and much convenient way of doing the propagation than cuttings, because you can actually store the plant materials for several years. And then I think that Luis has seen yesterday, told you about the pair breeding program that is developed after collecting seeds from different crosses of virus species. And the seedlings were grown in the field and selected by its vigor and non-chlorotic growth, like the ones that you see here, green in between the yellow ones. And then in vitro, what we do is the same thing as we did with that apple tree. You know, we cut the tree, collected the shoots coming from the rootstock, because the graft point was all the trees were cut under the graft point, of course, and rooted and produced thousands of plants. Okay, and in vitro one can see that the individuals that have physiological characteristics that solve the chlorosis, one can identify them as being the ones that are able to acidify the medium much more than the rest. Plants are propagated and grafted with commercial varieties and then they are evaluated in the commercial level. Okay, and then the other sort of technology is the generation of new recommendations. And in this case, the embryo rescue is used to rescue the embryos that are not fully developed when the fruits are ripened. These seeds will not germinate in soil. They were degraded by fungi. And as you can see, here's the process and the germination in vitro, and then the acclimation and the production of plants, viable plants that grow in the greenhouse and they are planted in fields and then selected by their fruit quality. We produce about 10,000, well, in total, 10,000 trees a year, considering the IRTA's breeding program and some private companies. We are working with peaches, nectarines, and also apricots. Apricot is the apricot breeding program is from south of France. These are some examples of peaches that have been selected and registered from our breeding program. Okay, and then comes the generation of more genetic recombination by double haploids. For those that don't, and polyploids too, but mainly double haploids. For those that are familiar, this is useless. For those that you are not, and then this could, I only pretend to clarify a little bit what is a double haploid. The double haploid it uses the gametes, the male or the female gametes on their own, so that after meiosis, and when there is recombination in the plant, each gamete has only one set of chromosomes, and the techniques to produce a diploid or haploid plant allows this set of chromosomes to double, or first of all, allows the regeneration of a plant from the gamete, and then in the process of regeneration produces a complete homozygous plant. As you can see, in each one of these cases, the plants have a complete homozygosity because they come from one gamete. Okay. The usefulness of this is the if I was going to make it simple, a double haploid is like a clone for the fruit tree, because the fruit tree relies on the clonal propagation. The vegetables rely on the production of pure lines, as if it was a clone. Okay, and then the pure lines are crossed and produce the commercial hybrids. But indeed, what is important for the breeding program is the availability of selected double haploids. So if one breeding program would have, for example, 5 or 10, the best selected double haploids during its years of work, this would be the most appreciated material, and then produced. Excuse me, I was interrupted. But and then they use those few double haploids to cross between them to produce the hybrids, and each year incorporate 2 or 3 double haploids to the crosses. This is produces the production of pure lines by double haploids reduces the time considerably, because by selfing and selection, one requires about 9 years or 10 years of work, while it's only required 18 months of work by double haploids. All right. This is a case of the beautiful case of the cereals. I only want you to take a look at this. This is the anther, and then the release of the embryos from the inside. These are the embryos and the plantlets growing out of the pollen. Okay. And the plants germinate and go into the greenhouse to produce the seed. One double haploid is the stable production of seed from microspores, in this case. This is the case of pepper. As you see here, excuse me, not pepper, the eggplant bud, the microspores that are selected when they are mononucleated. And this is the anther and the callus growth from inside the anther. The production of embryos, the embryos that grow into plantlets, and the plantlets that are grown in the greenhouse. so that there is a collection of seed. One double haploid is uniform in growth and in fruit shape and in all the phenotypic characteristics. As you see here in this row, there is a uniform height, and it's different from the rest of the plants that are after this, or the ones that are next to it. This is in the case of pepper. The production of anthers is guaranteed in a greenhouse, or it can be from a field. And these are the mononucleated macrospores as the right stage to do the anther culture, and the embryos that come from the inside of the anthers. Embryos and plantlets that grow to the greenhouse, and the production of the first fruits. And the seeds that are collected from these first fruits produce homogeneous, uniform, and self-maintained lines. Imagine that if this were an apple, it would be self-maintained by seed, which would be much easier to propagate than by cuttings. In the case of pepper, we had some selection of lines, double haploid lines that had a higher content of the red pigment that is called trans-cataxanthin, and these were sent to Zambia for the production of pigment. Of course, when one has a line that is interesting for some aspect, the maintenance of this is just a question of self-pollination, because all the plants that coming from a double haploid are equal to the parent. In the case of, this was up to here, the production of double haploid lines from pollen. In the rest of species that I will show you, the technique is the induction of parthenogenic embryos. Actually, apple is a species in which the parthenogenic embryos have been produced in a low frequency, because actually, as you know, each fruit has a few number of seeds, and the frequency is much lower than in the case of melon and all the vegetables. No? But as example, this is the flower. The pollination is done with gamma irradiated pollen, and then about 3 weeks after pollination, it is harvested. Of course, you have a fruit full of seeds, and then you have like 1,000 times more chances of having an embryo, a parthenogenic embryo, in a single fruit of melon than in an apple. The one here is that is filling the whole seed is a zygotic, while in here under it, you see this is a small white embryo. It's a parthenogenic embryo. The species solved the problem of lacking of a zygotic cross by producing an embryo that, of course, in natural conditions, it will not survive, but it's coming only from the female nuclei. And then this embryo that we can observe it by different ways can be rescued is again another embryo rescue process, and produce a plant, and the plant is haploid. And then when the plant gets propagated in vitro, because the in vitro conditions are favorable to get the plants grown, it can be doubled by a chemical treatment. And then the plants are brought to the field for selfing after acclimation. And in the greenhouse, the plants produce fruits from which the seeds are harvested. And then we have a double, from each fruit, a double, if we conserve any cross, of course, by bagging the flowers, we produce a melon from which all the seeds are equal, And when sowing those seeds, the plants will be equal between them and equal to the line. This is the watermelon. Of course, here we have a difficulty, that is the number of seeds. One could think that it would be the same for a fruit tree, but it could be solved also. And in the case of zucchini, it's the culture of the ovule by itself, not the induction of parthenogenic embryos by pollination with irradiated pollen, but only the harvest of ovules from the zucchini fruit, sowing them in a medium and producing plants that grow to the greenhouse and produce the fruits, each one producing a double haploid line. Okay, we had been involved, as you can see from the years that shown under it, we have involved for so many years and so long ago in the production of commercial hybrids of these species. Also in asparagus and carnation. And in tomato, we crash onto and I have to say, we failed. And as I have failed so many labs, and still they keep trying it, because if one solves this, it saves many years. And this could be an example of a species in which the process have found a great difficulty, and it could be applied to any fruit tree. No one is trying to do it in a fruit tree because of the amount of time, uncertainty, and probably money, but and the sector of production has other things to solve. Okay. There has been no possibility of applying any of the techniques. I only want to show you a thing in tomato, that is this thing. When crossing a plant of tomato with a species that is more close to eggplant, one has embryos that are small and are possibly parthenogenic embryos, but you are not able to rescue these in a conventional medium. And then what is done is to use a seed, remove the zygotic embryo, and place the parthenogenic embryo inside as if it was an horse culture. And from this, one can produce plants. As in the case of apple, I was gathering yesterday some information, and actually there were some parthenogenic work, as the case in melon, done to produce some apple, haploid apple varieties. Then they were double to diploid, and they were fertile. And there is also some work in androgenesis, so antherculture, done in Japan with this variety. Is just here the variety? No, under, well, we probably don't see it. Well, I don't see it here. Okay, these are a lot of work, but it has not ended up with a production of any commercial variety, but they have used it for molecular breeding, because it's very useful to have a completely homozygous line or clone. Okay, let's go on. These are the polyploids. The polyploids is they are considered useful for the production of seedless fruits, but they are also useful to produce more vigorous plants. It's useful for vegetables and ornamentals. And some, as I said in the beginning, some selected pair varieties in the field are not diploid, but they are samploidy, polyploidy level. As I said, and now we are switching to the development of a tool, because we spend so much time in the manipulating the plant material in vitro in order to get the objectives that we want, that we developed a temporal immersion system bioreactor that is named Green Tray to do the same work. So all these 3 lines of work could be done in this bioreactor. And it's this thing does, maybe there is too much noise, I don't know, probably reduce the volume of it. The thing is that the plants are inside of this recipient, and you can get all the plants out of the recipient, okay, and move them. And you don't use forceps to do that work. It's as simple as moving a drawer out of its place, no? And then this makes this instrument useful and different from any other bioreactor that exists in the market. In this case, let's see if you can see this one. The thing is that it's open and you can, is it moving or what? No. Ahora, okay. Is it, the recipient has 2 racks in which you could place the seeds or the cuttings easily. And in this case, we get another of these recipients and we cut the shoots with the scissors. We don't need then scalpel or forceps to do this work because they are dropped on the tray, as simple as that, okay? And actually, if you had a tapper under it, you could use the cuttings to root them directly in a greenhouse. All right. Sorry. We have them placed in models and that can be moved to, this is a plug and play module of 7. And we produce vegetative growth out of rootstocks. This is another one, another plant material. And the interesting thing is that you can cut, as you have seen, with the scissors and then put them back inside of the green tray to make them sprout again. And how many cuts can you do in a plant material? It depends on the genotype. This is a root pack, a pear, and another rootstock of plum, apple, rooted plant, apple plants, cannabis, Agrosulaceae. And actually this recipient can get the environmental conditions that you want. In this case, they are aerated and the humidity is dropped down to 60 every 15 minutes. So the plant gets to transpirate much more and uptake solution from the roots and make assays of, for example, this ability of different rootstocks to absorb calcium, and then select those plants that are able to absorb a higher calcium and transport it to the variety. In this case, is a selection to a biotic stress, which is the bacteria in fire blight, Erwinia millivora in apple. As you can see, the disease that is developing the shoot, and as I see here the reaction of a susceptible variety, one that is intermediate and a resistant one, or tolerant. Get the data out of it and being able to show which one is more tolerant than the rest. Okay. And I just want to show you here that the consideration that the selection in vitro with sugar, as done up to now, is much less reliable than doing it without sugar, because I didn't say it, but the aeration goes along with the use of a nutritional liquid medium without sugars. This is, as we have seen, more reliable having results applicable to the field than the in vitro growth. Okay, so the final consideration is that we are developers of techniques to recover, to preserve, generate, and propagate plants. And actually, we are at the service of broader objectives. Production for the production of plants in agronomic and industrial value. Okay, we have been working during the years, many people that are not here. This picture was taken actually 10 years ago. And we have had quite a few students and people in practice from all over the world and from Europe. All right, and that's it. This is a national park here in the Pyrenees.